DNA replication is a fascinating process, and it describes how life is able to store and copy information so that our cells are able to divide and so that we can continue to live more than just a day. And so I think it's really cool stuff. And what I'm going to do in this video is try to hit the highlights of how your DNA goes from being inside of a chromosome into actually becoming a separate daughter strand, how the parent strand goes from becoming two daughter strands of DNA that can now become two daughter cells. And so let's get into it. The first thing that's going to happen is we're going to start out by looking at the chromosome. The chromosomes are, in humans, we have 23 pairs, and each chromosome will have these chromatids. The chromatids are the larger regions, and typically you'll call them sister chromatids if they're together. And the chromatids are linked together via a structure called a centromere. And if you were to begin to take apart these chromatids, what you would get is some kind of substance, a larger kind of structure called chromatin. And chromatin is composed of tiny little beads, relatively speaking, that are wrapped many times in the DNA, the double-stranded DNA. And so histones are the proteins that make up those beads that are wrapped in your double-stranded DNA. And in this case, we're looking at the parent strand because we're about to perform replication. Now, as we'll recall about DNA, if we were to now look at a single double strand of DNA, we're going to realize that it's held together entirely by hydrogen bonds. These hydrogen bonds form between the bases that exist within the nucleotides, and we will always find purines bonded to pyrimidines. The purines, you can think of it as pure as gold. Purines are adenine and guanosine, and the pyrimidines, you can think of cut the pie, are cytosine, uracil, and thymine. And so you will always have an equal mixture of purines and pyrimidines in DNA. Now, if you were to zoom out a little bit at this point and watch what's going to happen next, if we were looking at a microscope, what we're going to find is you're going to see a little bubble form in your double-stranded parent DNA. And what that means is that you're observing the origin of replication. So this little bubble is referred to as the origin of replication. And in eukaryotic cells, in human cells for instance, our genomes are composed of about 300 billion base pairs. And so it's not uncommon, you're going to find many origins of replication because your genome is incredibly large. We have so many base pairs that we need to copy that you can't just have one event happening. So we've got many simultaneous DNA replications occurring, but for the sake of learning it, we're just going to look at one instance. And so what we're going to find if we took a snapshot of your DNA in a cell as it's dividing is that at time zero, we're going to have this little bubble. And as time progresses, we're going to see two replication forks for each bubble that we see. And these replication forks are these things that I've drawn arrows to right here and here. These replication forks move away from each other. And we also need to make a note how DNA is comprised of two anti-parallel complementary strands. What that means is that we have one strand. If we look at this picture here, we've got the blue strand running 5' to 3' in one direction, and the red strand running in the exact opposite direction, anti-parallel to the blue strand. Now, what these primes refer to is the carbon numberings on the ribose sugar that actually makes up the nucleotide. If we were to zoom in and look at the actual atoms that make up these nucleotides, what we're going to find is something that looks like this structure here. We're going to have a base, such as A, T, G, or C, and that base will be bound via hydrogen bonds to the parent's complementary base. So if we had adenine as our base 1, the parent's complement would have to be thymine. And adenine and thymine will form 2 hydrogen bonds together. So these 2 molecules are going to be joined in this sequence. What we find if we number the carbons is that on our 3' carbon of ribose, there exists this hydroxy group. And what we know from organic chemistry is that this hydroxy group will function as it can function as a nucleophile. And so the only thing that DNA polymerase will do in this perspective is facilitate a nucleophilic attack of the 3' hydroxy oxygen onto a phosphate or a phosphorus atom in an adjacent base. And so what would happen is, let's say that the next downstream base pair and the parent was cytosine. And if it was cytosine, the complement base would be guanine. And so we would form the H bonds, but we still haven't stitched together the actual sugar phosphate backbone that's going to hold the DNA together. And so what will need to happen is that DNA polymerase will come in and it will cause this 3' OH to attack this phosphorus here. And the special thing about this phosphorus is that it has a partial positive charge because it's surrounded by so many oxygen atoms, which are very electronegative. So a nucleophilic attack will occur. The 2 electrons on this oxygen are going to form a bond to this phosphorus. And what's going to happen in turn is the electrons are going to get kicked off. It's going to be a substitution reaction. And we're going to also form a pyrophosphate, which is just this leaving group here. But after that happens, we will have fully created the sugar phosphate backbone, and we can move on. Now, that's the biochemistry behind it and some of the arrow pushing mechanisms. But what's happening here, if we were to get back onto a higher level, is we're going to have a leading strand and a lagging strand. The take-home message from this organic chemistry is the fact that you can only build the complement strand in the 5' to 3' direction. Why? Because we need this 3' hydroxy group to attack the phosphorus atom on the next nucleotide. So the cause of that is we're going to get a leading and lagging strand. So as you're watching your genome replicate itself, what we're going to find are, as the bubble continues to grow, things referred to as single-stranded binding proteins that will attach themselves onto the actual DNA. They're abbreviated ssBP. And these single-stranded binding proteins will function to protect and stabilize the DNA, because the DNA on its own might stick back together and reform the hydrogen bonds that the 2 DNA helicases have been unzipping. And as they are pushing forward in opposite directions from each other, we're going to have the DNA polymerase. We're going to have 2 DNA polymerases in this field of view that are going to be actually building on these base pairs. And so if we were to make some room here, we'd find is that we'd have 1 DNA polymerase, and the thing with DNA polymerase on this one, if this was the 5' and 3' direction is that it can continue to build in this 5' to 3' direction very nicely. The problem is on the lagging strand, so the blue is the leading strand. On the lagging strand, there is no 3' OH that DNA polymerase can grab onto. So as DNA polymerase on the lagging strand adds on and stitches together the sugar phosphate backbone on the complementary strand, it will eventually run out once it reaches the end, because DNA polymerase here will have to be operating in the wrong direction. Draw this. So the two strands are anti-parallel to each other, and this will cause us to have problems, and this is resolved via something referred to as Okazaki fragments. Okazaki fragments are little sections of DNA that exist only on the lagging strand because of the fact that you cannot build in the 3' to 5' direction. And because of this, you need to use DNA ligases to stitch these fragments together. Now, for the sake of an introduction, I'm going to cut this video here. But a final note here is, if you're interested in how these Okazaki fragments are being formed, we need to have RNA primers present on our single-stranded parent DNA in order for DNA polymerase to bind to it, because DNA polymerase needs a lot of help. It needs those RNA primers, and it also can only work in one direction. So final take-home message is that we need something called primase to add the RNA primers to the DNA. And after the DNA has been replicated, the RNA primers are removed and switched back to the proper DNA, and they are stitched together using DNA ligase so that we have a nice continuous strand. And I hope you guys find this useful. Let me know if you have any questions, and thanks for watching.